This moves us on to the third portion of how information flows through your nervous system. And this one is related to neurotransmission, the chemical signaling process that occurs between cells. This is also the primary way in which neurons are interconnected. At the end of an axon, there are bubbles called vesicles. These bubbles are made from fat, which is the same material that makes the cell membrane. That's an important point because these vesicles are going to merge with the cell membrane itself later on in the process of neurotransmission. Now, these vesicles, which are essentially fat bubbles, are filled with neurotransmitters at any presynaptic neuron. Now, the location between two cells where they are connected always involves a presynaptic side, which is the side that's beginning the signal, then the gap, and then the opposite side, which is the postsynaptic side. The presynaptic side typically is the axon, and the postsynaptic side is typically a dendrite. There are alternatives to this story, but that's the most basic type of synapse that we're going to talk about. An axon on one side and a dendrite on the opposite side. The axon always being presynaptic and the dendrite always being postsynaptic. The vesicles that are filled with neurotransmitter in the presynaptic side are pushed against the cell wall once the action potential has reached the end of the axon. So there's actually a series of biochemical steps here that are not very relevant for us in psychology, but they generally involve the release of calcium, which pushes the vesicles against the very end of the axon itself. Because the vesicles are made from the same material as the cell membrane, as they land in locations called docking stations, the vesicles are torn open and all of their contents spill out into the gap between the cells. That material, the neurotransmitter, is essentially thrown out into the gap because as the vesicle is torn open, it is stretched and it essentially works like a catapult to push the neurotransmitter in the direction of the next cell. After the neurotransmitters cross the synaptic gap, they land on receptors. You've heard of receptors before, but what receptors actually are large protein molecules that stick out of the dendrite. So the neurotransmitter, as you might imagine, has a shape that will fit well into the receptor itself. And when it does, the combination of neurotransmitter and receptor will twist and cause a sodium channel to open up. There are a variety of charged particles that can go into the dendrite on the opposite side. Most likely, the charged particle that's going to go into the dendrite will be sodium. And you may remember sodium from chemistry class. It has a +1 charge on it. Now, earlier in the story, many slides ago, I mentioned that dendrites typically bring charged particles through themselves up to the soma. And we're finally reaching a point in a story where the whole story is actually wrapped around itself. So we're actually getting to the point of what's actually happening just before charged particles go into the dendrite. And what that is the activity of the cell on the presynaptic side. It releases neurotransmitter, which crosses the synaptic gap, lands on the receptor, and causes a channel to open for a charged particle. The most likely charged particle that will go in fact, the one that we will only generally refer to in a general psychology class will be sodium. And sodium is always positively charged. And from that point, you can actually go back to the story I've already told, in which the positively charged sodium ions go up the dendrite and into the soma. If you cross a positive 10 millivolt voltage within 1 millisecond, then you will have sufficiently cross the threshold to generate an action potential at the axon hillock so that a positive 40 millivolt action potential will come down the axon and cause the vesicles to be pushed against the inside of the end terminal of the axon, thereby releasing the neurotransmitter into the gap, crossing over, landing on the receptor, and then opening a channel on the dendrite. For our purposes now, we're talking about the neurotransmission process. So we've covered how the neurotransmitter, which is stored in the vesicle, is released from the presynaptic side, crosses the synaptic gap, and lands on a receptor on the dendrite. Once that combination of receptor and neurotransmitter have been held open for a while, the neurotransmitter will snap off. And because it is on essentially one side of an alleyway, when it snaps off, it only go back in the direction of the presynaptic cell. When the neurotransmitter reaches the presynaptic side, there will be openings in the cell membrane to reabsorb it. And that's called the process of reuptake. Reuptake is simply the name by which the neurotransmitter that has been released from a presynaptic terminal has now returned to the presynaptic terminal and is gone back inside. But neurotransmitters are typically inside of vesicles. So when during the process of reuptake, the neurotransmitter returns into the presynaptic terminal, it will be without a vesicle. And in fact, the vesicle that had originally held it has already been destroyed. It essentially has been absorbed into the cell membrane because, as I said, the vesicle is made from fat, as is the cell membrane. And so when the docking station rips open the vesicle, it will absorb the vesicle itself. So what will have to happen is a brand new empty vesicle will have to be generated inside of the axon. So during the process of reuptake, as the neurotransmitters are returning inside the presynaptic terminal, already constructed empty vesicles will be in there floating around and absorbing all of the free-floating neurotransmitters, and it will refill itself back up. At that point, the entire sequence of events is essentially complete. And so you are in a position now to make sure that the presynaptic cell can actually send another signal across the gap. So the presynaptic axon sends its neurotransmitters across, they return, get taken back up, reabsorbed by vesicles, and now the system is reset so that it can fire again. In this image, you can see graphically what we've been talking about. Along the top of the image, you have two cells that are connected together. The soma is exaggerated in size just to identify its location, and inside the soma you can see where the nucleus is holding chromosomes. There are many dendrites connected to the soma, and then the axon, which is one long tube, and drawn in this case with myelin as an insulator along it. At the end of the axon, there will be a location where the tip of the axon will merge with the dendrite in a gap. And if we take one of those locations and blow it up, that's what we see on the bottom of the figure. So the presynaptic gap is on the left side of the figure, and the postsynaptic side is on the right side of the figure. Arrows on both the right and left side indicate the direction of movement of information. Okay, and I remember, I say information because the activity of the cell involves three steps. First, charged particles go into the dendrite. Secondly, an action potential, which is electricity, biochemical electricity specifically, travels down the axon. And then thirdly, a neurotransmitter signaling system operates in the gap between cells. So between every single cell Anywhere in your nervous system, this entire sequence of events occurs over and over again. On the left side of this figure, you can see the vesicles, which are drawn as large circles, and they're filled with black dots. The black dots are representative of the neurotransmitter that they hold. When the action potential reaches the end of the axon, it will cause calcium to be released from the back side of the axon, which will cause the vesicles to get pushed against the inside cell membrane. At certain locations, those vesicles will land on places called docking stations, which will tear open the vesicle and essentially catapult the neurotransmitter across the gap. Because the vesicle and the cell membrane are both made from fat, the vesicle will be absorbed into the cell membrane and it'll therefore disappear. Later on, an empty vesicle will have to be generated, which will become active and usable during the process of reuptake. As the neurotransmitter crosses the gap, the neurotransmitter lands on its receptor, which is essentially a large protein molecule sticking out of the dendrite, essentially like a hair. The neurotransmitter has a complementary shape to that receptor, and that causes an ion channel to open up on the membrane of the dendrite. The most likely charged particle that will go into the dendrite is going to be sodium, which, remember, has a positive charge. And why that's important is because to get the cell on the right-hand side of the figure to generate electricity, you have to get a positive 10 millivolts of voltage in there, a positive 10 millivolts of charged particles into the cell within 1 millisecond. And of all of the dendrites that are attached to one soma, you have all of these charged particles going in, and then you may be able to cross the threshold to generate electricity. If electricity is not generated at any one time, whatever signal was passing through that general area simply stops at that location, and it does not proceed any further. When the neurotransmitter that has landed on the receptor that's sticking out of the dendrite has been there long enough, it will eventually snap off, largely from the mechanical properties of the neurotransmitter and the receptor twisting together. But as the neurotransmitter snaps itself off, you see in this alleyway, it's got nowhere to go but essentially back in the direction of the presynaptic side. So as it does, it will eventually find channels that will allow it to be reabsorbed back into the presynaptic channel, in which case it will be a free-floating neurotransmitter. But then a brand new empty vesicle will have been generated, which will come by and absorb all of the free-floating neurotransmitter, and then that system will be reset and able to operate again. How quickly can this occur? About 333 times per second. So actually very, very rapidly. Now that speed is not typical. That's a maximum speed set by the biological parameters of the cell itself. And I can tell you that, but I won't go into much detail regarding why those parameters are set in that sense, because it's just another level of biochemistry that eventually you'll get to in later classes. Now consider the operation of neurotransmission and how it affects a variety of different things, such as the treatment of psychiatric disorders. It's very commonly recognized that there are drugs that treat depression and anxiety and other types of hallucinations or delusional thinking and so forth. And the medications that are given are always going to be functioning at the synaptic gap, just as we were talking about. That's the primary job of a drug, is to get into a synaptic gap and cause some activity to the receptor, either increasing its activity or decreasing its activity. So when you take a look at a neurotransmitter, obviously the neurotransmitter is going to have some particular chemical shape. And because the neurotransmitter at any one synaptic gap is going to have a complementary shape to fit onto the receptor in that same area, you're going to restrict the size and shape of the neurotransmitter to fit the receptor in that area. Now, what will happen in terms of drugs is that pharmacologists will come along and create drugs that will get into the brain and have a similar shape to the receptors that already exist. If you make a man-made drug that doesn't occur naturally in the environment, and you can use it as a substance to stimulate the receptors or even suppress their activity, then that's essentially how this process is going to work. You're going to create a substance that you may generate completely in the laboratory. You may derive it from plant material. Sometimes you hear about, venom or other substances from creatures have chemicals in them that can be derived out and turned into a medication. And that's the general realm in which you're actually working at this point. But one of the things about this that you have to be very aware of is that various areas of the brain control different types of behavior. And so the receptors in that particular area of the brain will be unique in that general pathway. So for you to be able to become depressed, you must have an area of your brain that controls depression. And that area has to be activated in a particular way. And actually, we know in terms of depression, you have to have reduced activity in that area of the brain. And so if there is reduced activity in a certain area, you can generate a molecule in the laboratory that you could give to a person as a pill, and that drug will get into their brain and then increase the activity at those receptors, because the molecular shape of that drug is similar to the shape of the receptors in the area that controls depression. And the same basic logic holds true for anxiety. There are areas of your brain that control anxiety. So if you find out what the shape of the receptors are in that area, or the shape of the neurotransmitters that naturally work in that area, you can then work in a laboratory to generate your own molecule that does the same thing. And you may generate a molecule that works actually more effectively than what naturally occurs, or you may even produce something purposely that works less well so that it functions as a drug with a very minor effect, which might be something that you would want clinically. You also can find drugs that will treat hallucinations and thought disorders in the same way. The same logic applies in each of these scenarios, so that the neurotransmitter that naturally functions at a receptor has a complementary shape to the receptor, and once you're aware of the neurotransmitter shape and the receptor shape, you can work in a laboratory to generate a new molecule which would then fit into that entire system so that it will become a functionally usable component for that synaptic gap. To broaden the story a little bit, we have to look at neurotransmitter pathways. And there's a new rule in this case that I have not discussed yet. With respect to neurotransmitter pathways, you find that cells in your nervous system have a tendency to only communicate with other cells that have the same neurotransmitter in them. So for example, dopamine is a common neurotransmitter that you find in the brain. It is most likely, therefore, that a cell that has dopamine as a neurotransmitter in its vesicles will most likely only communicate with other cells that also have dopamine. And so if there's one dopamine cell connected to another dopamine cell, connected to another dopamine cell, connected to another dopamine cell, you essentially have a dopamine pathway, because a cell with dopamine mostly only communicates with other cells that have dopamine, and that forms essentially a chain of dopamine cells. And that's what would be called a neurotransmitter pathway, or in this case specifically, a dopaminergic pathway, because it's a pathway that involves dopamine. Now, of course, cells that have any one particular neurotransmitter will have to communicate with other cells that do not have the same neurotransmitter. Otherwise, you would limit all the communication in the brain to specific neurotransmitter pathways, which would really limit the amount of breadth of circuitry that you could generate in the brain. It's just that the locations where the activity from one neurotransmitter pathway connects to a neurotransmitter pathway of a different neurotransmitter are limited and exist in certain locations. They're essentially hubs where information is transferred from one type of circuit to another type of circuit. But generally from one hub to another hub, you'll just see a string of the same types of neurotransmitters residing along that entire line. Because we have this general rule that cells of certain neurotransmitters tend to communicate with other cells of certain neurotransmitters, and we have particular areas of your brain controlling certain patterns of behavior, what we have is a close correlation in the brain between a neurotransmitter type, a particular brain area, and whether or not the processing in that area is actually a sensory input process or a motor output process. So we're expanding on this whole sequence of events here. We're adding in the story of sensory input and motor output. Because essentially in your brain, you're either sending information into the brain from things you've detected outside your body, then you process it, and then you send information out. So in most cases, you're either in a sensory input pathway or a motor output pathway. Because of these other rules, neurotransmitter type, specific brain area, and sensory or motor processing are very strongly correlated together. So that in all the complexities of the brain, if someone were to say to you, I'm doing research on acetylcholine in the brain, that would cue you into what in general they're actually up to. Acetylcholine is a neurotransmitter that oftentimes is associated with a motor output pathway. So after your brain has essentially made a decision and it's going to control your muscles, it sends the information along output pathways. We haven't talked about those too much, but we're going to get to them. For now, know that the pathways out of the brain generally use acetylcholine as the neurotransmitter. So if a person told you that they were working on acetylcholine in the brain, they're probably working on motor output pathways, which would imply they might be working on diseases that include some type of motor impairment. For example, like Parkinson's disease, that would produce stiffness in muscles and tremor. So if somebody were working on acetylcholine, it might be that they're working on treatments for Parkinson's. They could also be doing advanced research in sports psychology and sports medicine to see whether or not they could increase the amount of activity in the outgoing pathways of the brain so that a person could run faster. Those are just other possibilities. But it's important to recognize the relationship between the neurotransmitter, the brain area, and whether you're talking about sensory or motor input processing. Because those three things being highly correlated together always give you cues as to what's actually happening in any area of the brain. It allows you to hear about dopamine or norepinephrine or acetylcholine, 3 different neurotransmitters, where in the brain you probably are, what's actually being processed, and the types of behaviors that are actually going to be modified by them. Now, we're not going to go through a list and work through lengthy lists to memorize these things. These are processes that you will see develop in your education over the course of coming courses. In this case, in a general psychology class, I would just want to lay out the general framework so that you can understand that there is a significant amount of logic embedded in the construction of the brain, so that even though it seems like a large mess at some times, if you have a few basic rules, the brain will actually make quite a bit of sense. Now let's take a look at this question and see how it relates to the things we've been talking about. Drugs tend to affect localized areas of the brain rather than neurotransmitter pathways. And the answer there is absolutely false. Drugs tend to affect neurotransmitter pathways, not localized areas of the brain. This is actually a very common misconception that, say, for example, a person has anxiety and they take anti-anxiety medication. That medication that they take, the pill that they take, goes into their stomach, right? They swallow the pill, goes into the stomach, gets dissolved, and gets into the blood supply. As the drug washes around the blood supply, it's not going to do anything out there. The anti-anxiety medication has to reach the area of your brain that controls anxiety before it can actually have its effect. So what will happen is the drug just washes around the blood supply, and then based off of the chemical properties of the drug itself and of the circulatory system in the brain, the drug will eventually start to diffuse into the brain. Now, the anti-anxiety medication may diffuse into areas of the brain in which there are no controls for anxiety, in which case that drug is essentially going to do nothing. But if it diffuses into the area that controls anxiety, then it will decrease the level of activity at those synaptic gaps and thereby decrease the amount of anxiety that the person experiences. So as you might imagine, this being essentially a large-scale chemical operation, a lot of the drug that you take simply washes around your blood supply and really doesn't go anywhere. Then it may just get washed out into urine. Some of it will get into the brain, but it'll get into the wrong area of the brain. And if the molecule itself has a substantially different shape from the receptors in the area that it got to, it will do nothing, because it can't connect to the receptor. It can't attach to it, and therefore it's essentially inert. It doesn't do anything there. And then even a smaller proportion of all the molecules of anti-anxiety medication you took will, just through chemical processes, reach the area of the brain that it needs to get to, and that's when you'll actually have the anti-anxiety effect. Now let's take a look at kind of a broader story here, because one of the things we're actually doing at this point is building up the nervous system from the smallest building block. And hopefully you remember me explaining that the smallest building block of the nervous system is the single individual neuron. And a neuron has 3 components to it that are always relevant for us in psychology. The soma, which holds your genetic material, which modifies the production of neurotransmitters and the production of hormones, both of which modify your behavior. Secondly, there is the axon, which will generate action potentials. And third, the dendrites, which are going to bring charged particles into the soma and will ultimately determine whether or not a sufficient amount of voltage has come in to generate an electrical signal or an action potential inside the cell. Now, I mentioned earlier in passing that there are sensory input pathways and motor output pathways. And we're going to look at that a little bit more formally now. Sensory input pathways are called sensory input pathways or sensory pathways simply because they are pathways that start at some receptor that ultimately brings information into your brain. So, a very simple one, any touch. Anytime your skin is touched, there are receptors in your skin that detect the physical pressure on your skin itself. And the physical pressing on your skin will cause an action potential at the receptor. Once that action potential is generated, that action potential will go down its axon, neurotransmission will occur to the next cell in the line, and then if sufficient charged particles go up its dendrite into the soma, a new action potential will be generated that will go down the axon and onto the third neuron and fourth neuron and so on and so on and so on. And there literally may be thousands of neurons before you actually reach your brain. But that entire sequence of events will start off only because some external source of energy had impinged upon some receptor from your body. So of course there is touch, there's going to be sound going in your ears, light going in your eyes, there are going to be chemicals that will go up your nose or onto your tongue. You have the capacity to recognize the position of gravity, so a sense of balance. And in fact, it's very important to recognize now that of the sensory pathways that you have, if I were to ask you how many senses do you have, the most common answer people say is 5, but the correct answer actually is 6. Balance is your 6th sense. Balance has a specific receptor system on the cochlea of your ear, which is entirely distinct from the portion of the cochlea that's used for hearing. So to say that you have 5 senses is sort of old now. You have 6 senses. Balance is your 6th sense. And so what there is in terms of the nervous system is only 6 inputs into your brain. That's it. There are 6 sensory inputs into your brain, and that's the only way to get sensory input into your brain. So everything that human beings have ever done since the beginning of time, all knowledge, whether it's super advanced science like quantum mechanics or philosophical work that goes back 10,000 years, all of it has largely involved just the input from 6 sensory processes. Then there is one motor output process. So of the 6 sensory inputs that come in, go into your brain, run through a sequence of analysis and decision making, which we have not talked about yet. Eventually, some final decision about how to respond is generated, and then signals leave your brain that will go out and control your muscles to move your skeleton around so that you can respond to the world in an appropriate way. So you see a box of donuts across the room, so that as light bounces off of the donuts, that light goes into your eye, so you see them, your brain processes them, I'm hungry, and then your brain makes that decision to move your body and walk over to the box of donuts. So it's 6 sensory inputs, 1 motor output pathway. And that one primary motor output pathway, even though it goes to various areas of your body, is considered one pathway because it all operates from one standard location, and it all uses acetylcholine as the primary neurotransmitter in any of that output activity. What you have in between the 6 sensory input pathways and the 1 motor output pathway are brain circuits. And brain circuits are really primarily what you're interested in the field of psychology. The work in terms of sensory input is relatively simple with respect to everything else because they tend to be very simple pathways that run from a receptor up to the brain. The same thing is true with the motor output pathways, except that a final decision about behavior is made in one of the last sections of your brain, and then action potentials are sent down those pathways to control your muscles. Brain circuitry tends to be much more complicated. So instead of referring to the cells as sensory neurons or motor neurons, you're going to refer to them as interneurons. Interneurons simply because they're in between the sensory input process and the motor output process. Please be very, very careful here. The neurons involved in the sensory input pathway and the neurons in the motor output pathway and the neurons that are part of brain circuits that we're calling interneurons are all the same neurons we've been talking about since the beginning of the semester. They are the same cell that we talked about that has the dendrite, soma, and the axon in it, and all the operations that go along with it that I've been talking about in this particular chapter are the same processes that occur here. These names, sensory, motor, and interneurons, are simply names that refer to the functional location in the entire system. So that if you had, in terms of your house, electrical wires coming in, delivering electricity to your house from the city utility, and then other wires exiting your house that lead somewhere else, you could simply refer to the wires that bring electricity in as input and wires that run outside your house to maybe a light on your front lawn as an output pathway. The wire is wire. It's just copper wiring. There's nothing different about it. It's just in a different part of the broader circuit. And that's why you call these cells sensory neurons or motor neurons or interneurons. It's the location in that broad, overarching scale of the nervous system that actually gives it its name. But the cell itself is always the same. So when you take a look at this whole process that I'm describing, the input pathway and the output pathway, there are a variety of basic rules that you have to absorb to make sense of the brain. First of all of your sense reception is going to pass through unchanging pathways in the nervous system. That is, the pathway from a touch receptor in your index finger through the entire pathway up to your brain is the same pathway it was there from the day you were born until the day you die. That pathway never changes. The same is also true with the motor output pathways. From the portion of your brain that receives the final decision about how you should behave through the entire motor output pathway to your legs and feet, all of that pathway never changes over your entire life. It doesn't get faster, it doesn't get slower, it always operates exactly the same way. But there is a part that actually does change, and that's the interneurons. The cells, which remember are the exact same type of cell, but they're just functionally in a different component. They're in between sensory input and motor output. The interneurons themselves constantly rewire themselves. The circuitry of your brain, for example, is literally changing day by day. And let's contrast that with an integrated circuit from inside the laptop or the computer that you're using to watch this video. An integrated circuit made by the Intel company is one static device. Yes, it's very small. Yes, it's very complicated, but it does not rewire itself. Okay, the pathways in an integrated circuit remain the same all the time. Yes, they do have switches so they can change the direction in which electricity follows, but they don't rewire themselves. Now consider a brain. The interneurons in the entire circuitry of your nervous system literally rewire themselves. So if you try to imagine the pathways that are generated that we've been talking about from cell connected to cell, connected to cell, connected to cell, you have many, many dendrites, many axonic branches, and the pathway that primarily travels through your brain can change over time. And what's actually happening in this case is that the changing pathway that occurs through the nervous system is one of the primary determinants that is represented in your ability to behave in a new way. So your perceptual aspect of learning something new indicates that maybe you learn to catch a ball. Somebody throws a ball, you don't catch it well, you practice, eventually you learn how to catch it well. The speed with which you learn to catch the ball, how long it takes you to be able to do that, is in part determined by your brain's ability to rewire itself so that you can coordinate your visual impression of a ball flying at you and then coordinating your hands to bring them up to be able to catch the ball. That has somewhat of a biological limit on it. If the cells in your brain cannot speed up, then you simply cannot learn more quickly. Of course, there's a variety of different ways to manage the speed or how slow somebody learns based upon the original arrangement of cells in the brain itself. But this is actually one of the parameters that you see, that there is going to be an upper limit with how quickly you can actually learn anything, and that is largely going to be determined by how quickly the cells in your brain can rewire themselves to generate a new circuit to represent a new pattern of behavior. That ability to rewire itself is called neuroplasticity. In fact, neuroplasticity involves a broader range of things than just rewiring of the brain. The term neuroplasticity is kind of a catch-all phrase, really, that refers to the nervous system's ability to adapt. So the adaptation of the nervous system oftentimes refers to new learned patterns of behavior, but it also refers to the production of new cells. So for example, if a cell is damaged and dies off, whether or not that cell can be brought back. We'll see in a short while that there are going to be different areas of your brain that will fix themselves better and other areas that you won't. And you have an intuitive sense of this already. In adults, brain damage and spinal cord damage tend to be fairly permanent. Not exactly permanent, but fairly permanent. And that's because your brain and your spinal cord don't really have the capacity to repair themselves well. So the aspect of neuroplasticity that exists there, even though your brain may rewire itself, if cells die in your brain, the brain and the spinal cord generally do not produce new cells as a replacement. However, if you chop your hand off or chop your leg off in an accident, you can get yourself over to the hospital and have your hand or your leg sewed back on. And then eventually your hand or your leg will work again once the doctors line up all of the nerves so that you can have control from your brain, run through a circuit, back into your leg or back into your hand. The cells inside of your limbs do repair themselves. And there are significant differences why these occur, and we're going to be looking at that in a little bit. For now, let's take a look at a question related to everything I've been talking about. Which of the following is not affected by brain plasticity? And the correct answer there is E. All of the above are affected by brain plasticity. Any new memory you form requires interneurons in your brain to rewire themselves to establish the memory. Anytime you learn a new pattern of behavior, like you've never ridden a two-wheeled bicycle and then you learn how to do that, that requires interneurons in your brain to rewire themselves so you can balance yourself on a two-wheeled bicycle. Repair of damaged nerves also involves brain plasticity or neuroplasticity, right? Neuroplasticity, brain plasticity, those are just different terms which mean the exact same thing. Your brain's ability to rewire itself or generate new cells. So reacting to any and all environmental experiences will include neuroplasticity, because for every experience you have, you are going to learn something new. That may not be obvious right now, but that's going to be a lesson from future chapters.